So first of all, I'm going to talk a little bit about how NIPT works. I'm sure some of you know some of this already, but there's a couple of interesting points perhaps that will be that might be of interest to you. So the first thing is we often talk about fetal DNA in the maternal circulation. That's the first lie. It's not fetal DNA at all, really. The DNA that we find associated with the fetus in the maternal circulation comes from the placenta. Now, as the placenta and the fetus are derived from the same fertilized egg, it's exactly the same genotype most of the time, nearly all of the time. We're going to come back to that. But when people talk about circulating cell-free fetal DNA, they're really talking about circulating cell-free placental DNA. And around about, on average, 10% of the DNA in the circulation comes from the fetus, and about 90% comes from the mother. But that is quite variable, and it's an important variable in terms of test performance. Now, as you'll be aware, the cause of Down syndrome, which we use as an example in this talk, though there's clearly other trisomies we can screen for at the same time. The cause of Down syndrome is an extra copy of chromosome 21. So if the fetus has Down syndrome, it follows that the relative amount of chromosome 21 to all the other chromosomes in the bloodstream will increase. And here's a few sums that talk about the level of increase that we have to detect. So this is a karyotype of an individual with Down syndrome. You can see that down here, instead of 2 copies of chromosome 21, there's 3 copies of chromosome 21. Now, chromosome 21 is about 50 megs long, and it represents around about 1.3% of the human genome. So if you do the sums and think, well, let's imagine this 10% cell-free DNA sample comes from the fetus. So in a Downs-affected pregnancy, instead of getting 1.3% of the DNA in a sample coming from chromosome 21, we'd actually get 1.3 for 90% of it, and 1.3 times 3 over 2 for 10% of it. The 3 over 2 is because we have 3 copies of chromosome 21 instead of 2. And the number comes out is 1.365. So the technical problem narrows down to, if we could tell the difference reliably between 1.3% and 1.36% of chromosome 21 DNA, then we would have a reliable test for Down syndrome. For a while, people worried that there wasn't going to be very much cell-free DNA in the blood, and in the 90s, in fact, there was considerable debate about which was the best way to go, because there was kind of 2 approaches to non-invasive prenatal testing. Some people were following the cell-free DNA route, whereas the alternative technology was to look at circulating fetal cells. But if you look at this little calculation I've put together here, you'll see why cell-free DNA has become so popular. So in a mil of plasma, when we get a mil of plasma, which is the cell-free material from the blood, we find about 10 nanograms of cell-free DNA. And 10 nanograms is about, divided by 3 picograms, is about 3,000 genome equivalents. So in 1 mil of plasma, we have about 3,000 genome equivalents. Now, one of the very convenient features of a cell-free DNA sample is that the DNA is already fragmented. Those of you who understand about next-generation sequencing will understand that you need to normally need to fragment the DNA before you sequence it. But of course, with cell-free DNA, you don't need to do that because it comes ready fragmented. And the average length is about 150 base pairs. Now, the human genome is 3.3 billion base pairs. So if you do the sum, you'll find that in 1 mil of plasma, you get about 66 billion fragments of DNA. So no shortage of DNA fragments in a mil of plasma to work with. And that compares and contrasts the number of circulating fetal cells in 1 mil of blood, which is quite variable. But let's say on average, it's about 6. So with fetal cells, you're dealing with very rare events, but with cell-free DNA, you're actually dealing with very common events. There's a lot of DNA in there. And then as DNA analysis technology improved, and particularly with the advent of next generation sequencing, it means that cell-free DNA has emerged as the preferred technology. So I know that some individuals are still working on circulating fetal cells as a technology, but really all non-invasive prenatal tests nowadays are more or less done using analysis of cell-free DNA.